Hello friends, good afternoon. How are you? Hope all are fine. Welcome to Sam Biology class. Today we shall discuss about paper electrophoresis. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Paper electrophoresis was introduced by P. Koenig in 1937. The use of filter paper as the stabilizing medium is very popular for the study of normal and abnormal plasma proteins. Papers of good quality containing at least 95% of cellulose and with a very slight adsorption capacity is suitable for this purpose. Chromatography paper is useful for electrophoresis. It needs no preparation other than to be cut to size. So in this technique, paper is the supporting medium. Under the influence of the electric field, charged sample components migrate through the paper to the oppositely charged electrodes at different rate based on their electrophoretic mobility and in turn are separated into distinct zones. Hence, paper electrophoresis is a type of zone electrophoresis. Paper electrophoresis is performed either in vertical mode or horizontal mode. Both are equally viable and the choice depends on our personal preferences. This figure illustrates the principle of horizontal paper electrophoresis. The equipment for paper electrophoresis consists of two items. One is a power pack and the other is a electrophoretic cell. The power pack provides a stabilized direct current. It has controls for both voltage and current output. The electrophoretic cell consists of electrodes, buffer reservoir, a support for paper, a support for paper and a insulating cover and a insulating cover. Each buffer reservoir is partitioned into two interconnected sections. This is necessary so that any change in pH occurring at the electrode doesn't affect the buffer solution which is in contact with the supporting medium. Paper is the supporting medium. These are the two oppositely charged electrodes which are usually made up of platinum. The paper is placed on an insulating material. These are the two glass plates between which the paper has been placed. Prior to the start of the electrophoretic process, the paper is saturated with the buffer solution. Sample application is one of the most important steps in electrophoresis. The sample is applied either as a spot or as a narrow uniform streak. After application of the sample, the whole unit is covered by an insulating material In order to minimize the evaporation of buffer solution during the electrophoretic run and to assure electrical insulation. After application of the sample, a stabilized direct current is supplied. Under the influence of the constant electric field, the charged sample components will migrate through the paper at a different rate to the oppositely charged electrode, and ultimately they will be separated into sharp distinct zones. After electrophoresis, the paper is removed and is dried in a vacuum oven at 110 degrees centigrade if sample components are thermostable. If thermolabile, then the paper is allowed to hang and air dried. A record of the separated component is called electrophoretogram. After separation, next step is detection and identification of sample components. Physical properties of different biological compounds such as fluorescence, UV absorption and radioactivity has been exploited to detect various compounds present in the sample. First method is fluorescence. Staining with ethidium bromide, and subsequent visualization of the electrophoretogram under UV light will make DNA or RNA fluoresce, thereby facilitating their detection. Similarly, staining with fluoresceamine, or dansyl chloride has been used for the detection of amino acids, derivatives, peptides and proteins. Second method is UV absorption. Amino acids, peptides, proteins and nucleic acids absorb UV light in the range of 260 to 280 nanometer. This property has also been successfully exploited for the detection. Third method is staining. Staining with specific reagent is also useful for the detection and identification of various biomolecules. Another application of paper electrophoresis is detection of enzymes. In situ. For this purpose, a special staining reaction is used. A paper strip of the same size as that of the electrophoretogram is taken and is impregnated with a substrate which is specific for the desired enzyme. But the substrate is modified in such a way that it will react with enzyme to develop a particular color. The paper strip is then just approached with the electrophoretogram. After a particular time, two strips are separated. The position of the appearance of the color on the paper strip gives the position of the particular enzyme on the electrophoretogram. Another application is quantitative estimation. Another application of paper electrophoresis is quantitative estimation. A rough estimate of the amount of components present in a sample can be obtained by visually comparing the colors of different zones with standards of known quantity. To get a more accurate value, the components are eluted from the paper and are dissolved in suitable solvents. The color density of the solution is then measured that gives a fairly accurate estimate of different components present in a sample. Friends, this is all about the principle, procedure and applications of paper electrophoresis. For more updates, subscribe my YouTube channel Sam Biology Class.